what I'm going to do now is talk about actual, more practical aspects, or more rounded aspects of what I was just saying in terms of. And back to the diagram that Ava's already showed, and again, you'll probably see, foundingGIDE almost kind of has two parts. There's a part about the global community and how we work together and how we influence, funder, and set policy, and that's what yesterday we were talking about. And then there's kind of technical part where we try, within at very least the participating resources, to do proof of concept work as to what interoperability might look like. And that's kind of split, and also good discussed yesterday about the fact that splitting between preclinical and microscopy or bioimaging clinical, or however you want to call that boundary, a lot of downsides for practical reasons. The way we're going at the moment is to have two streams of work, one of which focuses on the microscopy side, one of which focuses on the preclinical side. We'll hear about the bottom one later. I will talk a little bit more about the top one, with the idea that we have some degree of kind of harmonization, or at least something across these resources that lets us define in piece of this puzzle to be able to work together. And one way I kind of think about that is it's enough shared information between the three repositories, which I mentioned on the last slide, so the SBD, the IDR, and the BioImage archive, that we can have some interoperability. And with definitions like some is there incredibly vague, though, to, in a sense, keep us honest, what would we actually want out of interoperability? A starting point would be search. Can we put together something that will let you search for images across all these repositories and get back enough information to be useful? So this is actually quite a challenging problem. It's a bit yesterday about, and for the last few days, about reason managing image data is really complicated. It's complicated because imaging, again, is many different subdomains, different instrumentation, different technical requirements, kind of some different requirements then on metadata, different file formats. That's hard. Doing this globally, it's even harder because trying to do interchange adds complexity on this. So what typically happens at the meetings, and it absolutely had here, is we take this problem, this very difficult problem, and we make it worse. And the way we make it worse is by saying, okay, we don't just have to solve the problem managing imaging data. And Jan, who is now not here, so I can blame him, he's the person that kind of raised this, but it's obviously incredibly important point. We need to be able to link images to things on images. We need to be able to link to other molecular data and beyond. So this is a theory put together as a publication a little while back, showing how we might have a microscopy image, but we might also want to link it to, for example, an electron microscopy image. Lucy was giving some great examples yesterday how important correlative imaging is, but might also be able to want to link to information about the genomic content of what we're seeing. We might to be able to link to literature references. So sometimes this is just images. So correlative is, at least the starting point is two different image types where you've got to register into the same physical space. Sometimes this is, as I was saying, images and things like gene expression data, in this kind of rather beautiful image from OME-NGFF's lab of limb development, where each point is there's 12 different pleasantly typed RNAs here, and you need to be able to relate things together. You need to be able to relate the image to what each of these things really means. But in some cases, and one that is kind of increasingly coming up and also came in theme discussion over the last couple of days, say biodiversity. And I put this figure from I think it was actually a blog post from GBIF, and GBIF stands for Global Biodiversity Information Facility, who provide sort of search and data resources focused on biodiversity. And in their kind of data model relationship thing, the central bit is very much around biological specimens, and the biodiversity will tend to be specimen-centric, a lot in the sense that comes out of museum collections, to one of the things you tend to start by looking at, because they're already there before you go out into the wild. So their diagram almost starts with specimens, and then it links to, for example, genomic information comes up in here in the bit that's called GenBank, and then there's other things about applications and taxonomies. There is actually basically images up here, and because this is 15 years old, the bit that's images Flickr Explorer. Those of you might remember back a while when Flickr were a big thing. So kind of images come into this and come in here, and we from the Image Archive side have started to notice this because people have begun to ask us if we could host sample photography, basically, for biodiversity samples as part of various biodiversity projects. So the big one in the UK called the Darwin Tree of Life, which is trying to sequence all eukaryotic organisms, but in the sequencing, and the sequence kind of is the big part. It's also useful to have sample images, but the samples, the images are kind of the major player here. They are something that needs to be linked to from most the main bit of the data, but we have a responsibility, if we can, to try to provide our data in a way that allows it to be linked to usefully. And another way to summarize this challenge, this came out of a, this sort of got into my brain as a workshop organized by Alexandra in the audience out managing data from a project called, or image part of data from a project called RIKEN, which is a big M-led project sampling coastal samples from water and land and do imaging, but also lots of sequencing and all sorts of problems. And actually, one of the visiting speakers was giving examples of the kind of questions that we might ask. So suppose we want to be able to provide the imaging component that will let us answer interesting questions about, for example, how mitochondria, maybe mitochondrial shape, mitochondrial function, some of the things you only get from images might respond to climate conditions. Okay. Is this still working? Good. Right. Okay. So we have to do a lot of things if we, in order to answer these questions in the long run. We need to understand what the terms, this is maybe where the terminology controlled variabilities ontology comes in. We know what mitochondria means, we know what a marine organism is, but we also link across domains because we're thinking about image data and climate data here. And these will never sit in the same sort of, you know, as we talk about, we won't in the long run have one single point just for look for images. We certainly won't have one single point for look for images and climate information. This information will be broadly spread. We need to be able to link it together. Okay, so lots and lots of challenges here. Big, complex environment, many constraints, many different participants. What's the solution? This is really a solution that maybe it's an approach. Let's start by doing something minimally useful and then make it better. And then we iterate. Maybe this is how we get to a solution. And this talk and the end of this talk, sort of part one of a theme that Josh will pick on after the coffee break. And Josh already talked about, Jason talked about yesterday, for those of you who were at the end of the EAE, about the OME-NGFF challenge, trying to get some standards, enough standards to have interoperability across, in this case, 7 different participating groups. And I think we're really successful. Josh will tell you much more about this later on and kind of show all the cool things where you actually look at and visualize the images. I'm going to focus on one very specific part of this, and it's the part of this that in a sense was kind of new to me, but I think also new to some of the community. And from here onwards, what I'm going to do is split out the bit that's to do with the actual image data itself, which is actually the bit that's really more rooted in file format. I separate out the bit that's giving us the information here about the metadata, in particular the metadata that enables, for example, these things about organism and imaging modalities filtered over here to work. So there's some stuff here that is doing something useful. What is it and what's it doing? In effect, what in this the diagram labeled crate metadata? To answer that question, I'm going to have to define some terms. And first of these is, what is this thing called Crate? And so Crate is a big enough project that they have nice illustrators who can make diagrams to help with this problem. Crate, and RO stands for Research Object, is an attempt to have a very, very general standard for packaging research data. So this problem of how you put together reasonably heterogeneous stuff together with metadata is absolutely not unique to imaging. It comes up in lots and lots and lots of different places. Where the data are really, really, really standardized, have been for a long time. For example, like protein structures, which sort of talked about as models at time for the PDB. This problem isn't really acute because there is a good standard that everybody agrees on. Where things might be a bunch of people's different ways to write down the data in CSV files together with a video and something like that, you can't come up with a universal standard. What you can come up with is a universal packaging mechanism and an agreed way to share the metadata. And that's what RCrate does. It tries to give a way that you can take about different things and you put them kind of in the same box. And what is in the box is basically just data and metadata. And again, it's rather like file format in that regard, but attempting to be extremely general. Again, I'm going to subdivide and talk about the metadata side. What in this metadata box? Well, the metadata is described using something called JSON LD. So JSON, just again, JavaScript serialized object notation, is basically a way to write down structured information. Like, technically, it's a serialization mechanism so that you get complex structures into a single file. So JSON lets you describe things. And that what makes it LD, and the LD stands for linked data, is something called context. And the context in JSON LD gives you a framework for agreeing on what the terms you're using mean. So lots of people could describe sample and have different meanings by the word sample. Context kind of forces you to at least state what definition you're using. And this turns into linked data, and linked data is something that's been around for a long time. If you want to learn more about this, and this is where, I don't know if you'll be able to see this if you take the photo. I'm not suggesting you try and look at the poster here, but there's a fantastic description of a lot of the terms I've talked about and why linked data is useful and the ways in which you can use it in bioimaging context, made by Suzanne Kunis and Julia Dole for a project called 3D Bio. And this is on the Nodo, and not only is the poster, but there's also a video you can find which will walk you through the poster, and that's really great. So I would suggest if you're interested, go and look at this later. And we'll make sure there's a slide sharing mechanism after this so that you can find this, the Nodo identifier. So this will tell you about linked data. And linked data is very, very promising for solving some of these problems about linking across and within domains. It has downsides. It's in some ways more complex. It tends to be less intuitive. It's less intuitive for everybody to work with in some ways. It's very often tends to be less intuitive for software developers who are used to just JSON objects, which I think is part of the reason it's hard to adopt. It doesn't solve all of the problems, but I think it's promising for solving some of the problems we have. So back to get really specific, what's in the RCrate metadata that was made or collectively made across the participants in the challenge? 2 bits, or in fact, 1 bit, but I'll talk about, in a sense, the context and the content. Really, this is just a single file. It's one text file you can go and you can play with and you can look at if you're interested. And again, it's useful to think about it in 2 components, the metadata. And when we're trying to decide, I mean, we're having discussions at the OME meeting in May about, what's the real kind of minimal stuff we should agree to put in this box, what to go in it. We were trying to make a decision as to what was a reasonable minimum. And the outcome of the decision is that we're trying to describe biological images, and biological images kind of have to have 2 things. They have biology, so some concept of sample, and they have to have imaging. So there's some concept of something that captured an image. And that basically gave us the minimal bits that were in this box. So there is some information that describes, in this case, pretty much just the biological organism of what we were describing, you get taxonomy, and then something that describes the imaging method. And that's kind of it for the most. And I'll talk about where we get the minimal at the end. The context, again, the details don't matter so much, but it goes with it. And it's kind of, if you go look at things at the top of the file, forces us to write down the way that we describe these things. And this is one of the things which I'll summarize in a couple of slides' time, I think was so useful about this exercise is it's very, very abstract. We start to talk about, what ontology should we be choosing, how should we describe these things. When you're actually practically, you have to come up with a file and make some decisions, and we did this. There were various choices. These choices are almost certainly not all right, but they're a starting point. And the other thing that's sort of not visible directly by looking at the file, but you can visualize either using line tools or by kind of pen and paper and thinking about it, is that these things are linked together. So the file also describes how all of these conceptual ideas link together. So this starts to put together all of the three pieces I was talking about in the introductory session, or the introductory talk, about the data model, the metadata, vocabularies, and how they're linked together. And again, it's super minimal, because that's all we thought we'd do at the time. There's something about the biology, there's something about the image acquisition process. Really, really minimal, but why is it useful? It's useful because it's enough. It's enough to do something. I think Jason was saying lots and lots of grants shut down because federation's impossible. We kind of we've done it. We've at least made approval starting point. It's also a starting point to be able to expand, so we can try new things on top of this, and we have something where we can say, okay, this makes what we have. Are we okay with that? We've got something working to be able to go back to that. It also means that we can build tooling on top of this. So I'll say in a moment, one of the things that's probably really needed next to think about search might look like. But now we've got enough information there and data there to start testing that. It actually forces us to build something that works. So we are, and as part of the foundingGIDE project, we're doing a lot kind of reviewing of what ontologies are in use, and more about later on the different resources. That will start to move towards decisions as to be what we collectively think about. But for this, we actually had to make all the decisions within a very short period of time. And that's really useful for at least, forcing it to get out of the loop of what should we do, what should we do, what did we do, summit, and make it better. And it also starts to, and one of the things that was really interesting about the exercise, starts to highlight where the existing ontologies and vocabularies maybe aren't enough. And maybe my preliminary intuition here is that maybe surprisingly, where there are things that we struggle to find things that to do with some of the things that are actually imaging specific, are about the details of the kinds of specimen preparation protocols that only come in imaging. For example, in where specimen preparation is incredibly important, but also very complicated. There aren't really enough terms in the existing ontologies. Whereas for things like describing people, describing organizations, describing quite a bit of the biological sample side, actually there are good things out there and we use them rather than reinvent the wheels. Again, the exercise helps us focus where it would probably be more useful. Last slide, where do we go from this? I think there are different directions from what we've got already, which is the useful kernel of interoperability. These directions, I think, they will start whole focus have to go in different areas, and they will also start to stretch. What I think we'll probably end up doing is we've had one standard, one kind of thing everybody adhere to. It's probably, for example, not enough to make a minimum viable all of the repositories, for example, accept incoming data in this form, because there's lots of things that we would need there. A repository, from the repository perspective, you need to know a lot more about, say, the people, the organizations, the funding, the sort of ground technical metadata. We probably, so we need more to make it workable for repositories, but it's not necessarily what everybody cares about. We'd also need to think about where you go beyond minimally most useful. So there's a great, when we were kind of there in May, talking about is the NCBI taxonomy of the biological organism all need, and Jason made a big point, okay, you know, how do you distinguish between, is it human tissue, is it human cell line, is it a human organ? These things are all really different. Yeah, they absolutely are different, and we need to think about how we'd encode that, but you can't put the cell line as a mandatory field in this thing, because then the people that are doing tissue imaging will say this makes sense. So we need to test out, and there are ways to do this in linked data, so we'll need to test those out. We also are very invested in working with other repositories, and there have been a number of discussions before, especially I think for people who are looking to set up repositories about how to make them compatible with this. And the other thing, and actually probably the thing we want to do first, because it's part of the project and therefore we have to do it for the funding, so show how that search works. So different directions go on. And it might be interesting kind of see some of the discussion in the breakout session about how we take this forward collectively and what it means, and you know, is this the right approach? And this is also where I stop. So my timing is just about right, except the question part. Except, thank you. This is part one of the thanks. The reason this is part one thanks is because the people I'm thanking there are my immediate team. But the NGFF challenge is much, much, much wider than that. And there's part one, which Josh will talk about later on. And then perhaps at that point, we collectively thank much wider group of people who are involved. And thank you all for listening.